Morning, everyone. So I will continue showing trees, but not trees of trees, as Chris just showed us. I will show you trees of apes and humans. So the title of my talk today is What Genetic Changes Made Us Human. And I'm trying to argue that we are a very paradoxical ape. Before we start comparing humans to other life forms, I'd like to start out with the building blocks. You all know that you refer back to your DNA. It's in his DNA, it's in her DNA. Without DNA, there is no life. All life forms on the planet share these nucleotides. There's a few viruses that run on RNA. So nucleic acid is the stuff of life where the information of life resides. We all know that you need proteins that are encoded by DNA. But unfortunately, just nucleic acid, DNA or RNA, and proteins does not make a life form. If you want to build an ape or a human, you need other things. You need lipids. Cells are surrounded by cell membranes. They're out of fat. Fat is not encoded in the DNA. Fat comes in via the diet, can be modified by proteins, enzymes encoded in the DNA. But fat is one of these parts of life that comes from the outside as part of the ecological niche. And forgotten by many people are sugars. Sugars also are not encoded in your DNA. They come in from the food you eat. You can modify them using the enzymes that are proteins that are encoded in the DNA. But sugars are metabolically encoded. They're not encoded in your DNA. So the famous tenet of molecular biology that really founded all of biotech, the idea you can take a piece of DNA, let's say encoding insulin, and put it in a bacterium, and from then on, the bacterium is going to make insulin for you, the very same insulin that your pancreatic islet cells make. It's fantastic. But it's a very simplified view of life. If you want a living organism, as I mentioned, you need lipids and you need sugars coming in from the environment. And every cell is really a dense package of nucleic acid, the DNA of the genome, wrapped in fat, the nuclear membrane, surrounded by an ocean of cytoplasm full of lipids, sugars, and proteins, which itself is again wrapped in a layer of fat, which is the cell membrane. And for unknown reason, every living cell in this world is surrounded by a very dense layer of sugars, the glycocalyx, or the sugar coat, that defines the identity of the cells. It tells you whether you're still stem cells, whether you're going to become a sperm, an egg cell, a skin cell, or your eye. You're looking at me right now through a thick layer of MUC6 glycoprotein, which renders your eyes impenetrable to bacteria. But as you look at me now, you're looking through sugar on the surface of your eye. So these cells together with all these molecules can build an organism. And of course, there's input from the environment, and then you get your organism. But how do you make the next organism? You need a mate. So these are two apes in San Diego here making the next generation. When primates such as ourselves that have to have, our reproduction is only sexual. We can't go and bud off like a tree can or some bacteria can. We have to find a member of the opposite sex or a clinic. If you want to reproduce with a member of the same sex, you can have some help, but you'll have to cheat in a way, because to make the next generation, you need an egg cell made in a female and a sperm cell made in a male. Now, if you think back when you were a sperm, and each sperm contains half of your complete genome. The genome is the full collection of the DNA that makes you. Our genome has about 3 billion letters, so each sperm packages in it 3 billion letters of DNA, and the hopeful sperm will meet the egg that carries another 3 billion letters of DNA. And to put this into scale, I unwrapped some thread. So at this scale, the actual sperm is so small you can't see it. It contains about 1 yard of DNA, all the DNA in a sperm. At 1,000x, it's about 1 kilometer of DNA. That's what you're looking at, the thread you see up there. The same is true for the egg. And then you get a diploid cell, and your body is made up of diploid cells that carries 3 billion letters from your father, 3 billion letters from your mother. And of course, each one has been reshuffled between the DNA of your grandparents. And those cells divide and eventually make you. Now, if you take the genome of a great ape, and we now have genomes of all the great ape species and subspecies in hand for several individuals, and compare that to humans, you actually find there is quite a bit of difference. The difference, if you look at it at the scale of 1,000x, is about 5%. So that brown thread in the middle, that would be the amount of DNA letters that differs between the genome of humans and the closest living relative, the chimpanzees. So it's not that famous, oh, you 99% chimpanzee. That is still true for genes that code for proteins, but those only make up 2% of your genome. Most of your genome is made up of DNA that we're not quite sure what it does. It's not simple junk DNA that is sitting there doing nothing. There's a lot of hints that it does interesting things. But it includes DNA that has been lost in humans, but is present in chimps, and DNA that has been lost in chimps and is present in us. And if you include that, the difference is about 5%. It doesn't change the fact that the old saying of there are great apes and there are humans doesn't hold up. Because if you take DNA information on great apes and humans, you find that humans and two species of the great apes, namely the bonobo and the common chimpanzees that shared a common ancestor about 1 million years ago, are more closely related to us than they are to any other ape. So the term great ape is biologically nonsensical. It serves us right. We all know what we mean when we talk about great apes. And it was really, we were misled by the similarity between the face of a chimp and a gorilla. That anatomists would look at and say that both, they knuckle walk, they're not bipedal, they have similar sized brains that are much smaller than humans, so they must belong together. We know now, based on analysis of genomes, entire genomes, the 2 times 3 billion letters of many chimpanzees, 2 times 3 billion letters of thousands of humans, that chimpanzees and humans share a common ancestor after gorillas split off. There's 2 schools of thought. There's a lot of people who point out that really we're just another ape in many ways. And there's other people who point out, well, yeah, that's true, but we do so many things that no other ape does, no other species does. And depending on your inclination, you either espouse that, I want to lobby for the fact that great apes are just like humans, they should have human rights, and so forth. And other people say, well, it's, concentrate on the difference. That's where we learn about how we came about. So these 2 books would be an example of people arguing how similar we are. Jane Goodall being one proponent, and Frans de Waal. These would be 2 recent books that argue the other point. That, yes, we are very similar to the great apes, but that makes it even more interesting to ask what it is in our history that turned us into this paradoxical ape. So I showed you this huge amount of thread packaged into a tiny sperm or egg cell, really, really small. Pack 1 yard of yarn into this. How do cells do that? It turns out DNA is not a big bundle of yarn. It comes in chunks. Each chunk is called a chromosome. And chromosomes don't just consist of DNA. It's DNA that's wrapped very, very tightly around proteins called histones. And that's how cells can package this much DNA into a tiny little nucleus. When genes are expressed, it gets unpackaged. And then the DNA reading machinery can access the DNA and express the genes. So the first DNA that people were capable of seeing are chromosomes. They're called chromosomes in Greek. That means colored bodies. So people have been able to compare chromosomes between great apes and humans. And you have here an indication of the hierarchy of wrapping. That's the naked DNA. It's wrapped around these histones, which wrap around each other, which coil even more and coil more, and that's the chromosome you see. If you align the chromosomes of the great apes and humans, you see that they are remarkably similar, with one big exception here, is that our chromosome number 2, the second biggest chromosome in humans, still lives on as 2 separate chromosomes in the great apes. So sometimes in the last 6 million years, there was an event that fused 2 chromosomes. That's known for many other species. We don't know what it means. Quite embarrassing to admit that. Geneticists have no idea what the impact of the packaging in more or fewer pieces of DNA means. But it allows you to look at great apes and ask which chromosomal changes happened on which lineages. So this is a phylogenetic tree, not based on DNA, but based on how the DNA is packaged and how that package is changed. What's nice is it retrieves the fact that between humans and the 2 species of chimpanzees, you've got 3 really big changes that happened in humans, including that fusion of chromosome 2 I just talked about. So you can use chromosome changes to infer phylogeny, but you have no ticking clock like you have with mutational rates. But if you put all this genetic information together, you retrieve the phylogeny grouping humans as a sister group to the 2 chimps. If you now ask, where do humans live? It will give you some hints as to our crazy biology that has really departed from the biology of our closest living relatives. Living great apes need trees. They sleep in trees every night. Some of the big gorillas can sleep on the ground, but they all live in the tropical forests of Africa or Asia. Whereas humans, especially humans with money, you can see wherever these lights are lit up. And of course, the reason we have that photograph is that humans engage in space travel and can shoot satellites out into space, and then we can take pictures of ourselves and find out, well, look at that. The great apes live where you have fewest rich humans, which is part of the problem. They live in poor countries, and that's true for most non-human primates, which explains why many of these primates are in danger due to habitat loss, because desperate people desperately need the natural resources. So what is it about us that made this one species of great ape, I include ourselves into the great apes now, spread across the entire planet and colonize the planet? What about our behavior? What allows that? To illustrate that, I downloaded this photograph. This is the daily flights, global flights, human flights in planes, coded by flight time. You see the entire world is connected by these little metal tubes filled with people that fly from one continent to the next every day. But the fact that humans can do that, if you were to pack 300 male chimpanzees in a plane in New York City and fly them to Paris, how many do you think would survive? Not many, and we know because male chimpanzees are incredibly intolerant of males that don't live in their same social community. If they get a chance, they will try to kill them. So there's something about the human ape that allows 300 males that don't know each other to board a plane and fly to another continent without any lethal aggression. Unfortunately, there's an infinite list of behaviors that you could try to focus on. You know, pick your behavior. You could just take a dictionary, open any page at random, and you find hundreds of things that only humans do and none of the great apes do. Astronavigations, beekeeping, cycling, taking x-rays, engaging in yoga, or ziplining, for example. Now, we have a humiliating situation where people have studied human origins for many years, and we still don't have answers for some of the biggest questions, like why are we bipedal? How old is fire? Why is our brain 3 times as big as the brain of our closest living relatives? When and how did we become symbolic? How old is human language? We have no idea. And most of all, why are we the only species that is pondering all these other primates? Why aren't there ape anthropologists that are holding human skulls and pondering the differences? Now, Darwin and Huxley correctly suspected that the closest living relatives of humans would be the great apes of Africa. Darwin didn't have access to any great apes of Africa. He was quite familiar with a captive orangutan in the London Zoo that touched him profoundly because he spent some time in the cage with this young orangutan, and he was touched by how it seemed human-like. Huxley saw some specimens of great apes, but they had 0 fossils. I'm happy to report that we now have some fossils. In fact, we have fossils ranging back more than 6 million years with clear indication that these were upright apes. From the fossil record, we have indications, the oldest being Sahelanthropus chadensis, a skull badly crushed, but the foramen magnum, where the backbone gets into the skull, is under the skull. So this ape-like thing with a tiny brain, the size of a living great ape, was upright somewhere in Central Africa over 6 million years ago. After that, we have many different species of upright apes, some of them still with feet like that, later with feet like that. For a while, 2 coexisting apes, one walking like that bipedally, the other one walking like that, leaving footprints 3.5 million years ago, still with tiny brains. And not until about 2 million years ago, the brains suddenly get really big. And there is indication that these ape-like things now can run and can hunt and become the top predator, the top of the food chain. And the rest is history. They left Africa, they colonized the entire world, and they left Africa multiple times. Now, just to, as a disclaimer, most of these fossils would fit in one pickup truck. So it's not museums full of fossils. They're very precious fossils that thousands of paleontologists spend years finding, but they're quite informative. Unfortunately, for most of these, we have no DNA, but this has changed very recently with the advent of ancient DNA. We now have the paradoxical situation that for some fossils, All we have is a pinky bone, and scientists, geneticists, were able to get ancient DNA from this pinky bone, sequence the entire genome. Meanwhile, we have no information on their behavior. We don't know what their skulls look like. We don't know what their knees look like. But we have the full genome. We have some guesses at what pigmentation they had, whether they had blue eyes or not, and so forth. But we have no anatomy. So it's a very interesting situation to be in. We have ancient DNA and ancient genomes for a lot of other species as well, not just hominids. But that ancient DNA has kind of painted a very interesting story of human evolution, namely that there was a recent move out of Africa that is younger than 100,000 years old. And these recent modern humans that arrived in different places in Europe and Central Asia mixed with the people that were already there. But they did so in a very, very limited way. But we can prove in your DNA that you have some chunks of DNA that come from Neanderthals or these Denisovans, which is the group of humans in Central Asia. So using this actually allows geneticists to reconstruct mutation rate that is calculated by counting the differences that are not, the mutations that are not in something that is 40,000 years old, and compare it to modern DNA, allows you to calibrate the molecular clock. Recent advances in genome sequencing allow you to sequence the entire genome of a father, a mother, and their offspring. That's another way to get a mutation rate. And of course, you can compare chimpanzees and humans and count up the mutations. But these three estimates don't give you exactly the same mutation rate. So there's a very vivid debate among geneticists how to come up with the real mutation rate and whether that mutation rate could change over time. What I mentioned is we have clear evidence that when modern humans came out of Africa in the last 100,000 years, they picked up a very little pieces of DNA from these pre-existing humans. What is striking is that what little Neanderthal DNA you find in people in Europe and Asia doesn't usually contain genes. So there is indication that those moderns came out of Africa. There was some crossing with the local, more ancestral human species like the Neanderthals and the Denisovans, but the genes, the active pieces of DNA that code for proteins, were mostly thrown out, with a few exceptions. The most notable exception is that Tibetans were very recently shown. Tibetans are known to be the only human group capable of living at 5,000 meters, above 5,000 meters, and mothers can have normal birth weight babies. Han Chinese, who by now make the majority of inhabitants of Tibet, when they are pregnant, try to move to lower altitude because their babies will be low birth weight. But Tibetan women have perfectly normal weight babies. And that's because their babies can steal the oxygen from mom, even at 5,000 meters altitude. The gene that allows them to do that turns out to be a Denisovan gift that is not present in the Han Chinese. So there's a few exceptions where modern humans coming out of Africa have taken an adaptation from a Neanderthal or a Denisovan. And these exceptions include high altitude adaptation in Tibetans, some genes involved in pigmentations in Europeans, some genes involved in the skin, disease-resistant genes that allow people to fight off diseases that are not found in Africa, only outside, and lipid catabolism, how you deal with the fat in your food. That also seem to be from those. But in a nutshell, the take-home message is we are the last ones standing. Why is it that only one of all these species managed to leave descendants, where several of coexisting ancient species don't exist anymore? As I mentioned, all the early hominids are known from Africa, ranging from a 6 million year old Sahelanthropus chadensis in Central Africa, a whole collection of fossils dating back all the way to 5 million years ago that come from Ethiopia, which is also the place where you have the first fully modern human at about 160,000 years ago, and some very interesting early hominids, Australopithecines, between 3 million and 2 million years in South Africa. I'll show you some reconstructions. This is that badly crushed skull found by Michel Brunet in Chad, Sahelanthropus chadensis, nicknamed Tumai. These reconstructions are done by paleo artists and anatomists. They're mostly based on muscle attachment to the skull, which of course means we have no information about the type of hair, the pigmentation, the precise thickness of the nose and the lips. These things are open to debate. But these paleo artists are really good at taking ecological information into account. So this would be two different reconstructions of Tumai. at 6 million years, Ardipithecus ramidus, an absolutely amazing find by a team out of UC Berkeley and out of the Addis Ababa University. It took these people 9 years to write up their findings. They were published a couple of years ago in 9 back-to-back papers in Science. A 4.5 million year old female, ape-like thing, with a brain the size of a chimpanzee, clearly upright walking, but with feet like that, not knuckle walking, and lack of any sexual dimorphism in the canine that some people have taken as an indication for pair bonding. But most importantly, a pelvis that the top of it looks like a human and the bottom of it looks like a chimpanzee. So an absolutely, a fossil that sits in between. Ape-like in many ways, but clearly certain things have completely changed to accommodate the upright posture. Move forward 1 million years and you have the famous Lucy found 20 years earlier, Australopithecus afarensis, that was clearly bipedal, enough bones there to clearly, the pelvis is shaped like that, sidewards to hold the gut. The knee is absolutely adapted to hold vertical weight, and again, the foramen magnum, the hole in the skull, is under the skull, but a very, very small brain. Chimpanzee size and brain. About, this was I think 5 years ago, an Ethiopian paleontologist, Zerai Alemzeged, who's now at the California Academy of Science in San Francisco, found Dikika, also called Lucy's child, a 3.5 million year old fossil. Very, very interesting. You see a reconstruction of Dikika here. Interesting because there's clear indication in the skull of a slowdown of development compared to chimpanzees. This Australopithecine had a small brain, but it was maturing more slowly. It had a childhood. Chimpanzees go through development very fast and don't seem to slow down for input. Then another fascinating find, only a couple of years old, in a cave in South Africa, Australopithecus sediba, with, again, very small brain, tellingly a hand that for the first time shows this big thumb that looks very much like a hand that could make tools. Technology has advanced. You can see the skull is completely embedded in breccia, which is the limestone that drips in the cave. So you can't really open the skull and look into, but with CT scanning, you can actually represent the inside of the skull and get an endocast depiction, as you see. But 2 million years ago, you had much bigger brain things, Homo erectus, living outside Africa already, in Georgia. It turns out 5 skulls found in the same assemblage in Georgia are so different between each other, but they clearly lived at the same time in the same place, that it sheds a lot of doubt on these different skulls in Africa, each being given a different name. And that's, of course, a bias in paleoanthropology. If you find a fossil, which one would you like to find? The one that is our ancestor or the one that is the sideshow? By a bit less than 2 million years, there is this famous Nariokotome boy from Kenya, from Kubifora, that has very modern post-skeletal anatomy with a long body adapted for running, lots of evidence for stone tool use. That's the species that was the top of the food chain. It was hunting down a lot of grazing animals in East Africa. Later, you have things that are still very ancestral looking, like this Bodo skull, or Homo heidelbergensis, thought to be the common ancestors of Neanderthal and modern humans. This skull, interestingly, also has clear indications of defleshing. Somebody took an obsidian blade and cut out the eyes of this skull and scalped it and took the entire flesh off. We don't know whether this was a dead ritual or whether it was somebody's lunch. But clear indication that by then, human-like things, they were not modern humans yet, were very skilled with small blades, skilled enough to take out just the eye orbs from a skull. And then, of course, the famous Homo neanderthalensis, which was the only fossil that was found in Darwin's time. People disagreed what it was, whether it was pathological. Now we know it was a big population of individuals living from Europe all the way over to Asia. They were cold adapted. They were formidable hunters, but they did not have projectile weapons. They had big lances, but no projectile weapons. Depending on how you dress them up, you might not recognize them in the bus next to you. Depending on the tools they use, you might. Let's strip them down. This is from an incredible team of paleo artists in Holland, the Kennis brothers, 2 twins, that make probably the best reconstructions of ancient hominids. It's a modern human, the tall guy looking at the Neanderthal next to him. So one interesting thing that happened in the course of human evolution is that the development slowed down along the human lineage, with some indication already in this Dikika child. You can see here the different phases of development. What is amazing is that humans have a long survival after the reproductive period. That's especially true for females. And this childhood, where the growth of the body of a child slows down while its brain continues growing and while there is a lot of input through culture. And then with adolescence, possibly again, a slowdown before adolescence, and then a growth spurt that is absent in the chimpanzees. So it looks like our cycle, our schedule of development has actually allowed for periods where we can take in information and culture and then concentrate on growing and becoming sexually mature. So could it be that this growth schedule that's uniquely human among all the great apes is adapted for cultural opportunities, for nutritional opportunities, and is facilitated possibly by strong pair bonds between parents? Or more so by the fact that adults in most human societies, traditional societies, help the young of other adults, a phenomenon known as cooperative breeding or allo-mothering by both male and females. So what our growth schedule does, its delayed maturation allows for increased transmission of behavior and concepts. Human minds are effective copying machines. If you ask yourself, why do you speak the language you speak? Nobody really formally taught you that, your first language. You pick that up. We are born with a brain that is awaiting input in the form of language. And not providing that input is actually an insult to the human brain. So biologically, we are symbiotic with language. Our brains need input of language and are extremely good at picking it up without formal instruction. And of course, language is one of the things you don't find in the great apes. You find plenty of communications. People have tried teaching language to great apes. It turns out they were never able to utter human sounds, so people switched to sign language. The few great apes that after thousands of hours of training can learn quite a few signs never ask their keepers, so how are you feeling today? It's always, give me this, I would like another M&M, let's play with the blue ball, and so forth. So there is no conversation about today and tomorrow. What human language allows us to do is it allows us to cross-link. It's like an internet of minds long before Facebook and the internet, where you can learn from experiences that the elders have done. You can share those, you can scheme. You can talk about yesterday and learn from your mistakes and plan new mistakes for tomorrow. Another thing that language and our mind copying machines allows us to do is to pick up innovations. You only need one or two really smart people in a group and everybody else can copy it. Look at us. We all have the latest gadgets. We didn't invent those, but we fully benefit from the supercomputers in our pockets. That's because we're very good at picking up things that others have invented. Now, it's extremely hard to study the gestational development of a great ape. A Japanese team recently managed to do that because they have such a trusting relationships with living apes that they were able to convince this female chimpanzee to undergo sonograms during her pregnancy. And they measured the rate of development of the brain of this chimpanzee. This was the first time this has been done. And what they find is that in utero, before birth, the growth rate of the chimp brain, it slows down already. Whereas in humans, it continues. In fact, after humans are born, our brains continue growing at a fetal rate for a full year. So human mothers are actually outsourcing brain growth into that environment where everybody's talking to the baby, and there is culture and environment and behavior that the brain can pick up. Now, Filip Kajtovic, who is at the Max Planck Institute in Shanghai, he's a Russian trained with Svante Pääbo in Germany and now has his own institute in Shanghai, China, recently plotted brain growth over the ages with some of the behavioral changes you can see in blue here. And on the right side, you'll see some of the genetic changes that he links to changes in the brain. It turns out that what has happened in how we develop our brain is almost certainly a very big part of what makes us a paradoxical ape. And to convince you this is not just based on red and pink bubbles, here are some of the skulls used to calculate the brain size, and you actually see each data point of extinct hominid with the endocranial capacity, and along it mention of the behavioral changes. Note, we still don't know how old fire is, we don't know how old language is. So in terms of finding things out there in Africa that indicate to us that one of our ancestors was doing one thing as opposed to the other, it's tough, because these things don't fossilize. Many of these things don't fossilize. Now, this brain growth happened in the face of a major problem. The fact that you're bipedal forces you to change your pelvis in a certain angle and creates a massive constraint for birth. It turns out that chimpanzee mothers give birth very rapidly. It doesn't seem extremely traumatic. They rarely have help. Human mothers, on the contrary, need help. There are some cultures where the big goal is for a mother with her second and third child to just wander off and give birth alone. Most human cultures, mothers have help. Why? Because the baby has to rotate the head past these spines inside the pelvis and rotate it back. And the mortality associated with this head pelvic disproportion or cephalopelvic disproportion is huge. So something in our history in the last 2 million years has been pushing for these brains to be bigger and bigger despite a huge cost of losing both mother and baby. Interestingly, mothers stop being mothers and become grandmothers at some point. Another striking difference between great apes and us. You are hard-pressed to find an old female ape that is not ovulating. I just saw Sparrow again in Gombe this year. She's 53 years old and she seems to be menopausal. A huge exception. Most female chimpanzees continue having ovulations, getting pregnant until they're 50 and then they die. In human hunter-gatherers such as the Hadza of Tanzania, you see a grandmother here lifting a big rock to get at tubers, 25% of the adult population are post-reproductive females that have a huge role in transmitting behavior, concepts, language, rituals, and how you find food and you keep yourself alive. So that's another big change in the human life schedule, not how long do you stay child and small so you can pack in information, but stop reproducing because you're too precious. That's the famous grandmother hypothesis. You are kept in the population because you take care of the young ones without risking your life, continuing to risk your life with these dangerous pregnancies. Of course, among changes, there are very recent changes, the last 10,000 years with the advent of agriculture, where we start deciding which plants grow where, which animals we breed, and we start selecting for animals that give good meat and have certain traits, such as milk. Our diet has completely changed. And interestingly, with the change of diet, we have a massive change of the type of diseases that we die of now. It's quite clear that Homo erectus, this tall, running African hominid that became the top predator, massively benefited from the bone marrow it was getting from big bones, from the animal fat, from the red meat it ate. We would not be modern humans if it weren't for carnivory. Now we pay a very big price for the amount of meat we eat because, you know, it's a great idea to eat a lot of meat if you want to reproduce and be successful until you're 45, but if you want to live until you're 100, you pay a price for all that animal fat and the red meat. I thought I'd quickly address how some of these human behaviors have actually, via our behavior, changed our genome. And the most famous story is milk culture. There's evidence for an intense love story between some humans and cows. It's less than 10,000 years old, but there are more than 1 billion cows inhabiting the planet. So it paid off for the cows as well. There's far more cows than there would be if they had not been domesticated. Cattle domestication happened 3 different times, possibly in North Africa as well. What is important is those people who lived around animals whose milk they stole, very bizarre behavior. No other ape goes around stealing the mammary secretion of another species. We do so for camels, mares, cow, sheep, and goat. If you look at the genetics of being able to still digest lactose, a very simple disaccharide that mammals have invented to feed their young, most mammals stop being able to digest this when they become adults, after they are weaned. Makes a lot of sense because they will not encounter lactose in any of the food they eat. But those humans who live together with mammals whose milk they steal had a huge benefit if they could continue digesting the lactose as adults. This has exerted a selective pressure so strong as to select for 2 different mutations, one in Europe and in West Africa among the Fulani, and another one in East Africa, the people like the Dinkas and the Maasai who live with their cattle, that allow the adults to continue drinking milk. So this is an example where that cultural behavior that we mimic and we take in with our slowly and ever-growing brain has changed our own genes. And so there's a lot of interest in finding other examples like that, where human behavior is changing the genetics of humans. I thought I'd briefly go over the type of genetic changes that happen in evolution, and how these can be used to finger changes that have happened only in one in the human lineage. So I go by bigger chunks of DNA that change to smaller chunks of DNA. These include chromosomal rearrangements. These are usually tens of millions of base pairs that change. Segmental duplications. Your genome, which is a long text of DNA, has these chapters. And some of these chapters can copy themselves. And you suddenly find not just one chapter, but another chapter that looks remarkably like the first chapter. Those are known as segmental duplication. They are a type of massive genetic stutter of big chunks of DNA that can copy themselves and, like an accordion, make your genome bigger or smaller. There's a lot of evidence that these things have changed between humans and great apes. We have endogenous retroviruses. We have a big part of our genomes that are actually viruses that became part of us, that we have domesticated, so to speak. And there are some examples where you make some novel biological function, like human and great ape placentas are different from any other mammalian placentas because human and great apes, our common ancestor, used the retrovirus to make a better placenta. So there's all kinds of dynamic processes in the genome that allow it to change. And of course, there's tiny little differences like single nucleotide polymorphism where one letter only has changed between a human and a non-human. That's the case for the lactase persistent gene I mentioned before. It's a single letter change that doesn't turn off an enzyme when you're adult, and then you can drink milk and not get sick. Just one typographical change. People aren't even sure how many functional elements there are in your genome. It clearly, the whole genome is 3 billion letters, as I mentioned. There's between 23 and 25,000 genes, these telltale sentence that starts with a ahem, a promoter, that tells you that's a gene, then you find exons and introns. But these code for over 100,000 different proteins because you can edit the chapters via splicing, and then you can change the proteins by which lipids or sugars you attach to them. So even though you only have 20,000 genes, you have millions of parts that make up your body. There is promoters, which are places in the genome that attract the attention of the cells that come here and start reading this gene. Lots of those. There's a lot of alternative splicing where the same gene can give rise to 5 or 6 different proteins, depending which chapter you leave in or you throw out. There are 500,000 enhancers. Enhancers are a very metaphysical concept. They are chunks of DNA that can be far away from a gene, but they influence whether this gene gets expressed or not. There is a lot of RNA that never makes a protein, but it actually affects how your genome is expressed. These microRNAs target 60% of the genes. So if you're only interested in the gene, you have to be aware that there are little snippets of RNA that never become a protein, but they impact how much your gene is expressed, where and when. There are RNA binding proteins that orchestrate, once the message has been read off the DNA in the form of an RNA, it can be sequestered and held on for a while and then released to be expressed. So the expression of these 25,000 genes in each one of your cells is an incredible choreography where, depending on whether you are a cell in the eye, in the brain, or in the big toe, the same set of 25,000 genes gets expressed in very different ways and allows you to make very different cell types. What's interesting is I mentioned that the coding genes that code for all your 25,000 genes for proteins, there's another 2% of your 3 billion letters that don't seem to change much between chickens and primates. There are no genes there, but for whatever reason, these regions cannot be messed with during evolution, or you get a non-organism. A strong indication that what until recently people called junk DNA is not just junk DNA. There are some hidden important elements there. So there's a type of genome syntax. Rather than to think, okay, it's simple, it's DNA that tells you who you are, it's not that simple. Because you have chromatin remodeling. I mentioned DNA is wrapped around proteins. If you unwrap it one way, you can express some genes. If you unwrap it another way or don't unwrap it, you can't express the genes. Histones can be modified, so the spools, the protein spools around which the DNA is wrapped, they can themselves be changed by little molecules that are involved in imprinting that either shuts off a gene or turns it on. DNA itself can be annotated with little groups of methyls attached to the DNA that usually shut down a gene. There are these non-coding RNAs, a completely new field of research. And very importantly, there are RNA binding proteins. One of the most famous of those is a protein expressed in neurons that binds to 900 different RNAs that's involved in autism and Fragile X, mental retardation. So if we go back to the genome, and you remember I showed you these chromosomes that are chunks, the 46 chunks of DNA you find in a human, 2 times 23, and you ask, if you compare this to great apes, what kind of differences do you find, and where are these? What you find is that there are some places where there are copy number differences. Humans have so and so many copies of one gene, chimps have fewer. There's other places where chimps have more copies, and we have fewer. You can't simply express that in percent difference. That's quite different. It's not a strictly linear difference. There are novel genes. There are some genes that exist in humans that do not exist in chimps. There are pseudogenes, genes that have been shut off in humans, but they're still active in the chimps. There is a lot of evidence of positive selection. Genes that have changed, something forced these genes in the last 6 million years to change only in humans, whereas in the chimps and the great apes, they've been humming along just fine. This is an example of segmental duplication. A big chunk of millions of letters of DNA that for unexplained reason went berserk in chimps and gorillas. It's all over their chromosomes, and in humans it's just found on 2 chromosomes. And there's the reverse of this. So genomes are dynamic. Genomes are like these big texts, these big books that can have chapters copying themselves and pasting themselves in and out. And we don't really know what this means. We know that this can change how genes are expressed, whether or not they are expressed. I'll show you a few examples. This is a chromosome that has a gene, and that gene has duplicated twice only in humans. Chimpanzees don't have this. And people here at Scripps were actually could take this gene and put it into the brain of a mouse and ask, what does it do to the neuron of a mouse? What it does to the neuron of a mouse is that neuron makes more dendrites. It splits more and talks to more cells. Another great discovery is something called the human accelerated region, a stretch of 180 letters that hasn't changed between chickens and chimpanzees. And in humans, it has 18 changes. So it's something that has been left alone in all these other vertebrates, including birds. And humans have changed this like crazy. This is not a gene. It doesn't code for protein. It turns out it codes for a little piece of RNA that is involved in brain development. And so this is one of the places where there has been hot action in human evolution. And this gene codes for an RNA, and the RNA is expressed in the fetal brain of humans, which makes it almost impossible to do experiments in humans. But that's an example, and there is a long collection of these HARs. A recent review by Katie Pollard, who discovered, was a co-discoverer of these human accelerated regions, asked, what do these do? It turns out most of them regulate gene expression in the brain. So there's a lot of indications that the place where gene expression has changed a lot in humans is with regard to brain development. You can take these and you can put these genes, the human version, into a mouse and tag them with a little color reaction and find out that another place there's been action is in the digits. There's the opposite, where humans lack a piece of DNA that's present in all the other species. One of these lacking pieces is for the androgen receptor. It's responsible to make spines on penises, tiny spines that great apes have, microscopic spines. Humans don't have these. It also makes the vibrissae that humans have lost, the great apes still have. Another example is a tumor suppressor gene that is expressed in the brain. This gene allows cells to develop for longer. If you put it in a mouse, those brain cells in the mouse can divide longer than they could if that piece is not there. So sometimes losing a piece of DNA allows the organism to do something new. This is a study from Philip Kaitovich, who looked at a quite respectable number of great apes, brains, chimpanzee brains, human brains, and macaque brains, and compared the cerebellum, the back of the brain, with the frontal cortex, where you do all your higher order reasoning, asking, is there a difference in how these genes are expressed at different ages? It turns out he could show that human genes, the same genes that are expressed early in chimpanzees and macaques, are expressed late in humans. So it's a strong evidence for neoteny, this idea that you go through the same developmental program, but you slow down the program, and you end up with a different animal. People had long observed that a young chimpanzee seen from the side looks much more similar to a human than an old chimpanzee. So in the brain, the gene expression does the same thing. Humans seem to have a brain that can do novel things because they slow down its development. Very interestingly, one of these genes expressed is a gene that changed between Neanderthals and us. Another thing that might be very interesting for the younger ones among you, who is younger than 30 in the audience? That's good. It turns out your brain is not fully myelinated. Myelin is the wrapping around your axons that allows nervous signals to go faster. Myelinization is finished in great apes before the age of 12. In humans, it goes on well into the 3rd decade. So this is another example where we let our brain grow longer and we give it much more time to fully wrap up, because that's the opportunity where you can have input and then change your brain depending on the input. You can look into the brain, and one way you can do that now is with novel visual techniques like diffuser tensor imaging. This is, if you look at the kind of scary part of the brown brain cut up, is the arcuate fasciculus that's really big in most people on the left side, that connects you two language areas, the Broca's area and Wernicke's area. It's gigantic in humans, only on the left side. Those of us, most of us who are left lateralized, including left-handed people. It's really small in chimpanzees. This is probably one of the most extreme anatomical differences between humans and great apes. We have a superhighway of communication between the area that produces language and that perceives language that already exists in the chimps, but it's tiny in the great apes compared to humans. So neuroscientists are homing in on the parts of your brain where language is happening. And there's clear evidence that that part massively changed only in humans compared to the great apes. There's some very interesting gene mutations that have been found in humans who cannot properly learn how to speak because they have a problem with sequencing, how they produce sounds. One of these genes is FOXP2. FOXP2 is a transcription factor, so this is a protein that goes and binds DNA of hundreds of other genes and is part of the choreography of how genes are expressed. These two mutations that have occurred only in humans have been put into mice, and they've changed how mouse neurons develop. They've also changed how the mice vocalize in ultrasound. People have called this the language gene, which is ridiculous because language is almost certainly comes about by the interaction of a lot of different genes and the input. But it looks like this gene is part of a network of changes that has only occurred in humans. Another area, to get back to the first cartoon I showed you about every cell being surrounded by sugar, is a change that my colleague Ajit Varki discovered over 20 years ago, which is essentially a mutation that has completely changed the cell surface sugars only in humans and not in any of the great apes. This change happened over 2 million years ago. Since then, a lot of our biology of proteins that go about fingering the cell surface, the way your body knows that other cells are self, has a lot to do with the sugars that are on the other cells. And the molecules that finger these sugars are called siglecs. They're like immune globulin family genes that go around touching other cell surfaces. It turns out after humans changed their cell surfaces, they've also changed a whole collection of these siglecs because humans changed how their self looked, and they had to then adapt how to recognize self. None of that action is seen in the great ape. This is interesting because it includes a lot of pathways involved with diseases that are common in humans but absent in great apes. It's also interesting because it's true at the level of sperm. To return to sperm, you see a picture here of chimpanzee sperm stained with an antibody for this sugar that is completely absent from humans, but it's covering the entire surface of chimpanzee sperm. And so my lab is very interested in the question of whether if you go through a wholesale makeover of your cell surface, the price you might pay for that is that you become reproductively incompatible. Because these chimpanzee sperm to a female that has lost these same sugars look like non-self. And we can show that in vitro experiments. We can expose chimpanzee sperm to human serum and show that they get killed because they don't look right. So chimpanzee sperm would have not much chance in a female reproductive tract, an experiment that will never happen, luckily. And I'll end with a crazy way that evolution of genomes works, and this goes back to Dr. Will's talk about how pathogens influence how you evolve. Natural killer cells have special molecules involved in defining self and non-self, and you see here a collection of genes that differ between humans and chimpanzees, but also between individual humans. So those part of our genomes that change the most are the ones that have to do with surviving infection and dodging infectious agents that come in pretending to be self. So in summary, I think you have to take into account the syntax of the genome when you try to look for differences, and this will be online, so you can go through this at your own pace. I just noted a couple of recent examples for changes only in the human lineage that have to do with histone modification, with DNA methylation, with non-coding RNA or RNA binding proteins. There are examples for each one of these categories. And to go back to the big picture of what we want to explain, that's the collection of fossils we have over the 6 million years. We know that bipedality is very old. Bipedality is 6 million years old. How old is pair bonding? Some people are, propose it was a big part of the story. We are the only ape that lives in a big social group with sequential pair bonding within that group, but we don't know. We don't even know how old home bases are. When did we, as the only ape that uses a home base, settle down, at least for a while, use a camp? Some baboons do that, but no other great ape does that. Manual dexterity, some good evidence that 2 million years ago we were there. Fire, oldest good evidence, 1 million years, but we don't know. Very important because of cooking and how it allows you to use many new foods. How old is music? Is music the precursor for language, or is music a consequence of language? We don't know. There's all these questions of behavior that you only see in humans, all the way down to using shoes. There's some chimpanzees that put fibers around their feet when they climb on thorns, but they don't really have shoes. So in this interest of finding out what it is that makes us different, we have to keep in mind that we're looking over a huge time period that includes things like changing to bipedality 6 million years ago, to using full-blown language to engage in mental time travel and tell each other stories and ask questions, which none of the great apes seem to be doing. Archaeologists in Africa are finding very old evidence of things like beads and microblades. These things are clear indication of behaviors you don't see in any great apes. There is a report of chimpanzees using a spear that should be taken with a block of salt. They use a piece of wood that they jam down a tree to molest the little primate until it comes out, and then they eat it. The spears the Neanderthals were using 200,000 years ago were a massive thing, allowing them to kill big animals. But then, almost 70,000 years ago, you have arrowheads in Africa. Somebody invented projectile weapons, which might have totally changed the demands on your cognition. You have to predict where an animal moves so that you can shoot it with something that also moves, and so forth. These things leave fossils. We don't know what fossils to look for to know when we became symbolic. We have shared symbols now. We have personal names. None of the apes have names for each other. There's no way you can say, hey, did you see what Frodo did yesterday? They don't have names. How could you say, did you see what Frodo did? You have no language and no names. Kinship terms, very important in most traditional societies. You keep track of paternal and female relatives. Shared rituals, sacred spaces, group identity. You paint yourself, you laugh at the others because they painted differently or wear different stuff. Of course, these group identities help when you have intergroup conflict. I thought I'd throw in how difficult it is to track down some of the more complex behaviors. I mentioned that proteins are long stretches of amino acids. Sugars, polysaccharides are long stretches of molecules. Well, humans have long been using things to make fiber. The oldest fiber known, human-made fiber, is only 30,000 years old. But of course, with fibers, you can make amazing things. You can make nets, you can make baskets, you can make hunting machines, traps. None of these things would fossilize. Who knows? Maybe Homo erectus was using them 2 million years ago. You would never find them in the fossil record. I mentioned intergroup conflict. There's a lot of interest in what it is about humans that can make us so aggressive, because we stick together within groups, but we're very good at being nasty on outgroup. How old is war? Is it old enough that it shaped this human cognitive behavior? Some of you might be familiar with Steve Pinker's proposal that actually, if you look at the data, humans, if anything, have become less and less violent over time. But maybe it's not behavior. Maybe, let's go back to ecology, it's climate. It turns out there have been incredible changes in climate over these 6 million years of time that might have acted like a behavioral pump where you better be flexible, you better come up with a string or a new technology that allows you to survive. Very interesting ideas, but there's at least 2 species that are still around and didn't become the global dominating species. They include hyenas and baboons. Always lived where these human ancestors have lived. Baboons are very widespread, one of the few African primates that are not endangered, but they didn't become smart, they didn't develop culture, neither did hyenas. So climate alone will not do it. Maybe it's social factors. Darwin called blushing the most peculiar form of all human emotions. We have a skin, and if, like me, you're melanically challenged and you blush, everybody knows that you have some emotions. We read our emotions on our skin. All humans blush, but it's more visible in people who have less pigmentation. And I'd like to end on a note revisiting hybridization. It was great how some of you asked about hybridization in the trees. So we now have evidence that hybridization happened. It happened between modern humans and Neanderthals and between modern humans and Denisovans. It did not contribute many important genes. In fact, there is evidence that after hybridization, most Neanderthal genes were thrown out. But hybridization continues to happen in captivity. This is a hybrid bonobo chimpanzee. The father was a bonobo, and the father is still alive, is a bonobo, and the mother is a common chimpanzee. And she's grooming me. She lives in France. She's one of 6 sibs, has a brother who is fertile. And so with some colleagues, we've sequenced the genome of these individuals based on a skin cell, and found that, in fact, they are the first-generation hybrid. And we're hoping we have established stem cells from these cells that we can now study. What are further candidates that we should look at in terms, important part of our biology where we find differences? Clearly, things like cancer genes, because great apes don't get the same cancers as us. But the key thing is nobody can make sense of this molecular, the genome syntax. How is a string of DNA related to what happens in a cell that's 3-dimensional in time and space? We keep overlooking important changes that have to do with sugars, and this is me touting our sugar horn here. There is a sugar modification that affects a transcription factor called O-glucanac. Almost nobody looks at that. It's known from cell biology that stem cells and different stages of cells differ just by what kind of sugars they have on their surface. The extracellular matrix, the other half of your body that makes up your bones and your connective tissues, it's all sugar biology. We know almost nothing about differences between great apes and us. If you start cooking, one of the many things you change are the sugars in the food that you eat. And of course, the microbiome is established by the milk you drink from your mother, the microbes you live with, and these microbes are very important in digesting oligosaccharides. So I'll summarize by saying that I think we are, there is a paradox, because there is continuity. We clearly are an ape, but a very, very different one. The landscape of functional elements is huge, and it should be humbling to all the genome scientists. Say, oh, all we need to do is sequence all the great apes, sequence a couple of thousand humans, and we'll know what happened. Not at all. It will be decades before we know what happened. But we have good chancing. We have those very good candidates. We already know there is a lot of action in brain and development. We're enriched in changes that affect brain development. And some of these are actively being pursued now. And one sad thing is, of course, opportunities for studying great apes, their phenotypes, how they behave, how they develop, are rapidly disappearing. With the Chimpanzee Protection Act, it's become virtually impossible to study chimpanzees in the US, even when your studies are totally non-destructive, non-invasive. So the key things we need to keep in mind when thinking about how different we are these. And I'd be happy to take some questions. Thank you very much.